Dear colleagues, ladies and gentlemen, good evening from Athens, Greece. It is our great pleasure to welcome you in another Spotlight on Science Masterclass. Today, this masterclass is going to be really interesting because the topic is hot and timeline for all endocrinologists and scientists that have to do with metabolism. We're going to discuss about dual and triple agonists, their molecular mechanisms and the clinical impact. We have the honor to have with us two very important scientists from Germany, Professor Matthias Zoeb and Professor Timo Miller. They are really very specialized in the field, and they have contributed a lot in the development of the field. Please be free to ask questions throughout the masterclass. You can use the chat, or during the discussion session, you can raise your hand, switch on your camera, and speak. But it's another very easy way to do is to write your questions through the chat. We record the session. This is one of a lot that we already did. Most of you have already participated in many of them. So now I'm going to introduce our first speaker, who is Professor Matthias Zoeb. Allow me to say a few words about this very important scientist and professor. Matthias Zoeb was trained in medicine and endocrinology in Munich, in Germany. After a research fellowship at Eli Lilly in Indianapolis in the States, he advanced to research director and chair of medicine at the University of Cincinnati. In 2011, he was recruited back to Germany, in Munich specifically, and now he holds there a professorship, an Alexander von Humboldt professorship. Today, he is also CEO at Helmholtz Center Munich and vice president of the Helmholtz Association of German Research Centers. He has been awarded multiple awards, such as Schrödinger Prize, Paul Martini Prize, Jeffrey Harris Award, Karus Medal of the German National Academy, Ernst Jung Prize in Medicine. In a few weeks, he will become the first German to receive the Budding Medal of the American Diabetes Association. He holds an adjunct professorship at Yale University in the States and an honorary doctorate at Leipzig University. Professor Zoeb is an elected member of the German, Bavarian, and European Academies of Sciences, the American Society of Clinical Investigation, and the Association of American Physicians. Matthias Zoeb has unraveled fundamental gut-brain signals to discover breakthrough medicines, the dual and triple gut hormone multi-agonists. A first representative is FDA approved. Others are successfully progressing through clinical trials, offering more than 20% body weight loss and unprecedented blood sugar control. These new therapeutics enable the reversal of the obesity and diabetes pandemic, a scientific achievement previously believed to be impossible. Professor Zoeb, is a great pleasure and honor to have you with us, and we are looking forward to enjoy your lecture. Thank you.
Well, thank you very much, Lena, for the introduction. So let me start by thanking you again for the introduction and invitation. This is a great pleasure to be here. I've been a member of the European Society of Endocrinology for many, many years, and I've always enjoyed attending meetings and conferences. And I think these types of workshops, as we are participating in now, are a great addition to the program. So with that, I get started with the topic that you have given me, the discovery of dual and triple agonists. If I understand correctly, it will be Professor Timo Müller, who will be focusing on the mechanism more. So I tell you the story of the discovery and a little bit of what we think might be clinical impact of these compounds. Now, this can probably most suitable be started with a look back in history, because we now have more than 100 years since a fantastic breakthrough in metabolic medicine, which was the discovery of insulin. This was transformative because it turned a deadly disease into a chronic disease. Now, that said, 100 years of additional research, insulin is still used as a life-saving treatment, but it hasn't really solved the problem, has it? If you look across the world, there is massive continued additional increase of the global prevalence of diabetes, posing a lot of suffering and challenges to health systems and economies worldwide. So that discovery has saved lives and is saving lives, but it hasn't solved the problem. Now, the mechanisms how one has to do with the other continue to be researched, but we certainly all would agree on the fact that if there wouldn't be an obesity pandemic, sort of a pandemic in slow motion, then there probably wouldn't be a type 2 diabetes pandemic of these proportions. However, these two are really molecularly linked together. And obesity is not only a major pandemic driver of diabetes, specifically type 2 diabetes, it is also a deadly disease and something that causes numerous problems with several disease areas and in several organs. So you would ask, why don't we do anything about this? Well, this was sort of the question I started asking, and it's now frighteningly almost 30 years ago that I was a young physician working in the endocrine department of this hospital here, the University Hospital in central downtown Munich, which, by the way, is the director of this place now is, I believe, to be correct here, the president of the ESE, Martin Reinke. Back then, I was a young doctor seeing patients with diabetes and obesity and realizing how frustrating it was that, all you can tell patients mostly is, try to eat less and do more exercise. That is a mantra that goes back basically to Hippocrates, and it hasn't worked any better ever since for numerous reasons. One alternative, of course, was and is to basically call a surgeon and consider if a gastric bypass can be done, and that is a real option to prevent complications and morbid obesity, etc. But it is a highly invasive and irreversible and certainly not risk-free operation. So at that starting point, there was nothing out there in terms of obesity drugs, always this and that, but nothing that would go beyond, let's say, 2, 3, 4% weight loss, specifically when you consider what placebo effects looked like. And then there were always new drugs discovered that some of them would actually work, but they had side effects that were severe. And then drugs wouldn't get approved or were approved and then were taken off the market again. So there was nothing. There was a clear unmet medical need. And as a young endocrinologist to be, I didn't want to believe that with all the power we know hormones have, there's nothing we can do to come up with a therapy that, let's say, looks similar to medication for hypertension, right? Where you can, at least with combinations of 1 to 3 drugs, get control of most blood pressure situations. Now, this was an exciting time, and that coincided, and it had a major impact on my life and career. This was the discovery of leptin. Now, imagine a young endocrinologist trying to go into science, and here's the discovery of not only a new hormone called leptin that powerfully acts on the brain to regulate food intake and body weight. It is also the discovery that there is a new endocrine organ called fat, adipocytes. Something we basically looked at for many decades as sacks full of calories, and something that is not participating in system-wide communication. And here is this groundbreaking discovery by my friend Jeff Friedman, who really taught us about a new endocrine organ, a new hormone, and the first molecular basis for body weight regulation. That, in my mind, is the start of modern obesity research, molecular obesity research. But it also has been a major step toward destigmatizing obese patients that have many times by society been looked at as, well, you should just, exert more discipline, and it's your personal fault, and you are struggling with obesity, not recognizing that there are molecular reasons and genetic reasons that drive obesity. Now, when I was so impressed by this finding, I decided to go into research and went to Eli Lilly and Company as a postdoc fellow in Indianapolis, because that was the only place in the world where they could actually make soluble, stable leptin. And I wanted to learn more about it and understand it and see if there are other factors that are regulating appetite and body weight. And as a matter of fact, we then ran into the discovery that there is another factor called ghrelin, and ghrelin is the endogenous, if you so will, leptin opponent, something that comes from the stomach rather than adipose tissue. It's more of a short-term rather than a long-term signal, but acts at very similar locations in the brain in the opposite direction to increase hunger and appetite and body weight and fat mass. Now, it turns out that our hope that back then really existed, that leptin may be the end for the obesity epidemic, or ghrelin blockade may be the end of it, didn't pan out. There is something that you can summarize as leptin resistance. Giving leptin to obese individuals doesn't really help a lot, may actually hurt more than it helps in the end. There's high leptin levels circulating in obese populations. There's already low ghrelin in obese populations. It's tricky to block the ghrelin pathway, and it's also not very powerful in terms of outcome. We still learned a lot from this. We learned, for example, that the brain really matters for obesity. As a matter of fact, when you combine these findings of ghrelin and leptin acting in the brain with everything we know about genetics of obesity, human genetics, and Sadaf and Steve that you see here have really been pioneers of that field, also recognizing that the melanocortin receptor system is really important for obesity, for human obesity. You could sum up in one sentence saying obesity, human obesity, is a brain disease, because all these genetic operations that we know, the polymorphisms, etc., that are relevant for human obesity have something to do with either players, signals, molecules, or functions that happen in the brain. Now, knowing that, we thought, well, we have to somehow find ways to come up with new drugs that help us prevent, fight, overcome obesity, but we got to do so safely. And some things were tried with cannabinoid receptor interactions, etc., that turned out not to be safe. And to put it simply, our pharmacology wasn't and still isn't specific enough to allow us to go into the brain, into those places, specific circuits, specific astrocytes, specific neurons, where you're only manipulating metabolism and appetite and hunger and not affecting other systems, mood, cardiovascular, behavior, etc. So you cannot be effective and safe until now to manipulate systemic metabolism without doing side effects when you target the brain directly. But you could do so indirectly, we thought. So this was our consideration early on, that we said, well, there is Mother Nature's toolkit of peripheral hormones, such as leptin and ghrelin and some others, that are acting in the brain, knowing exactly where to go, having receptors only at places that matter mostly for metabolism, appetite control. Why not use that indirect way, powerful but safe? And it relieves us from the challenge to really come up with specific pharmacology that finds these cells in the brain that are scattered around all of the CNS. Now, how to do that? Well, there's so many factors that were discovered and so many pathways, afferent pathways after leptin and ghrelin, that it became hard to choose. And Translational pharmacology is a tricky thing. It takes a long time. You just have so many shots at the goal in a lifetime. So we had to have some guiding factors, and turns out bariatric surgery, gastric bypass, and so on was one of them, because there are changes that occur in hormones, endocrine players after a gastric bypass is done. They happen early, and they may have something to do with early metabolic improvements that even seem to occur before body weight is lost. So our thinking was, is there a functional role to these hormones? And maybe we can learn something from the patterns that happen before and after gastric bypass that guides us in selecting the right hormones to turn those into drugs that indirectly, safely manipulate appetite and metabolism. And learning from ghrelin and leptin, we were convinced it's a pattern that we have to generate. It has to be more powerful than a single factor. And so which combinations are those? You can imagine, how difficult that is, because there's so many combinations and variations. It's like, trying to get the lock for a safe open, or, doing a Rubik's Cube, all these combinations. So we went after that. And if you try to go after this efficiently, and you're an ex-physician and now pharmacologist, physiologist like myself, doing in vivo things, you need a chemist. So I teamed up with Richard DiMarchi, one of the world's leading peptide chemists, who had then already made drugs that were approved and helping patients in endocrine diseases, and insulin and others. So the two of us really sharpened that hypothesis and said, well, which combinations out of what I just said are the right ones to try, and went back between his chemistry lab and our in vivo lab with Timo and others to test and validate and improve and optimize. So we need a polytherapeutic approach. We want to use endogenous gut-brain signals, indirectly targeting the brain, and we need to target more than one human gut hormone receptor. This is, again, based on afferent, safe, effective signals, and it is based on the fact that we need more than one and want to go with these bariatric surgery influenced signals. We came up with interest in glucagon, and now there is a long history of glucagon. Actually, that's another centennial anniversary we're going to have this year or next year. The discovery of glucagon goes back 100 years as well. Now, as you know from textbooks, glucagon does a lot of things, but most importantly, or most memorably, it increases blood glucose. It's the counter-regulatory hormone. If it prevents you from going into hypoglycemia. But it is not everything it does. Glucagon does a lot of other things. It's influenced by many factors and situations. It helps with driving thermogenesis, helping to burn calories. It affects food intake, etc., etc. So glucagon actually would be very desirable as a factor to have in a multifactorial new drug, but you got to be careful that you somehow prevent it from exerting its diabetogenic liabilities. So how can we accomplish that? Well, the first thing is you look at that family and see, maybe combinations are possible out of that family that drove some of our interest, but then you have to make glucagon molecules you can play with, you can test, you can optimize, right? The endogenous glucagon, has a very short half-life, it's not easily soluble, etc. So we had to make new tools. And this is what Richard does. He understands endogenous hormones extremely well and can tweak them to become more like a drug, still be recognized by the body only as sort of a natural hormone, so not being attacked with the immune system, but more powerful, so you can really study its action profile much better. So some of the thinking that helps here is what we learned from the heli monster, this lizard here, where in the saliva there's a version of GLP that's called exendin that has the CX tail that you can also use to put it on other endogenous hormones like glucagon and make them more long-acting and soluble and powerful. And that was done. And here's an important Eureka experiment where we observed for the first time, I believe it might have been in 2006, that infusion of these kinds of more powerful glucagon molecules into a diet-induced obese insulin-resistant mouse changes body weight powerfully. It decreases it. These were implanted osmotic mini pumps, and comparable to a highly active GLP-1 receptor agonist, and GLP-1 receptor agonists had been known already, including their impact on food intake and body weight, which wasn't enough, but was something that could be used as a benchmark. So seeing these similar effects on body weight in mice with two very different hormones, with very different receptor populations, and thought to be action profiles, we thought, well, there's something we could combine. There's room for synergy, and that was then done. But I want to shortly stop here and remind you that this was going against everything that was believed in at the time. People definitely called us crazy and said, every pharmaceutical company is looking for a glucagon receptor antagonism. You want to look for agonism. That is dangerous and risky, and you shouldn't be doing it. You will never get funding. That was sort of the view of that at the time. We did believe it adds significant therapeutic potential, specifically when you combine it with something else, such as a GLP-1 receptor agonist. So next phase was, can we now come up with molecules? Can Richard in his lab and with his team come up with molecules that are activating both receptors, a dual agonist, right? That was achieved. Here is the paper that is now, 14 years ago that it came out. But of course, research started almost 20 years ago on this. The first dual agonist ever discovered, a glucagon GLP dual agonist. Now, with that, we were able to really drive down body weight with once weekly injections in mice that were obese, 20, 30%, much faster than we wanted, and probably much faster than would be safe in humans. Now, that was encouraging, but we were still nervous what would happen with glucose metabolism. Turns out nothing bad. Insulin levels normalized, glucose tolerance normalized. So it seems we are activating the glucagon receptor. We're giving glucagon here, and still we don't see hyperglycemia because we're improving body weight, we're decreasing body fat, we're improving metabolic flexibility, and GLP-1 with its powerful incretin effect and insulin sensitizing potential is overriding the diabetogenic liabilities of glucagon to a large extent. Now, we looked in glucagon receptor knockout mice just to make sure that there's really something in GLP-1 receptor knockout mice. It's really something there that goes beyond just a GLP. And as you can tell, we still see effects on body weight and body fat. By the way, there is different levels of glucagon receptor activation in these two compounds. The more glucagon we have in the molecule, the more we drive down body weight and body fat. And this is in mice that don't even have a GLP-1 receptor. This, of course, only worked in mice for now, but later was validated in humans. These are examples, and AstraZeneca and others, showing that this principally works in humans. There's now clinical trials on the way, phase 2, I believe, probably going into phase 3. And normalizing blood sugar, body weight significantly dropped. So this, in principle, works in humans, and actually also affects things like total cholesterol and triglycerides to an extent it was not previously believed to be expected. However, we got to put in our mind again the fact that glucagon receptors are on hepatocytes, and these dual agonists are targeting the liver a little bit more than probably anything does that is targeting GLP-1 receptors, because those are not on hepatocytes. And we all know how important the liver is for lipid and glucose metabolism. We also know that fatty liver disease is frequently a part of the metabolic syndrome and observed in obesity. Okay, so second point, dual agonism at the glucagon and the GLP-1 receptor outperforms GLP-1 receptor monoagonism. We wanted to do more, though, and we wanted to go a little bit a different path, just in case that, glucagon wasn't panning out going through phase 2 and phase 3 trials. After all, there's little known about long-term glucagon receptor activation, and knowing from a history of obesity research how many of these drugs have failed in the past, we thought it would be wise to have a few shots at the goal. So we looked at another approach that had to do with GIP. So another incretin hormone similar to GLP-1, another gut hormone, but also something that comes up when you look carefully at metabolic control systemically that is orchestrated by the gastrointestinal system, and also when you look at bariatric surgery impact. Now, GIP and GLP-1 have a history that goes back for a long time, and arrives today at a time where we again thought we can change the paradigm, because for a long time GIP was looked at as one of the bad guys in endocrinology and in metabolic control. Again, we had to make a GIP receptor agonist. The GIP receptor agonist that would have longer action and be soluble, and something we can work with, and not just have a half-life of a few minutes. And this is what Richard did. And just for you to know that this is always trial and error, right? So this is, you make compounds, you optimize them, you check them, and you see how they work at the mouse receptor, how much do they work at the human receptor, there are differences. So these things take not weeks and months, they take years. You've got to bring a lot of patience, and there's a lot of suffering involved in terms of seeing experiments that don't work. So not everything I'm showing you just worked. Most of it didn't work. You get to see the 1% of results in the end that were successful after many, many, many failures. So we found then, or Richard found, GIP analogs that would be powerfully working in vivo. They should work in mice that have no GLP-1 receptor, but they should not work in mice that have no GIP receptor. So they're specific and selective. And again, we observed that with long-term GIP receptor activation in mice that are obese, we could decrease body weight. Again, unexpected for everything you would believe back then in the field, a paradigm shift. This worked repeatedly, and again was confirmed in the appropriate knockout mouse model. So as a summary here, GIP receptor agonism, not antagonism, offers significant potential for the therapy of obesity. This is what we concluded here, went against everything that the field believed in at the time, but the data are friendly is what Richard used to say. We went with those data. Okay, now, again, can we combine this with something else that's powerful, like a GLP-1 receptor, and really break through that threshold of 5 or 10% body weight loss and go toward more like 20% body weight loss? This is the paper that shows that we were successful, published in 2013. This is the second dual receptor agonist class of drugs that we discovered and reported. And it's important because it's a dual incretin, a GIP-GLP dual agonist that is what tirzepatide is, Mounjaro, a drug that has by now been approved by the FDA and is the first in class and best in class out there today in the clinical world. So we saw that in mice, dose-dependent decrease in body weight, very impressive improvements in glucose metabolism. We actually tested that. We tested that in primates. We tested our versions of these dual agonists in humans, a clinical trial that showed it decreases blood sugar and body weight powerfully, dose-dependently in humans. And here are the data of the version that Lilly made later, of course, with additional pharmacological improvements and a little bit of a different balance. So we had, I think, almost 50-50 GIP, GLP, believing in the GIP agonism. Lilly even went further, and they have a 5-to-1 ratio of GIP receptor versus GLP-1 receptor agonism. And let me stay with this for a moment. That gives 22.5% weight loss in human obesity after 72 weeks. So just for you to put this in perspective, when not having been around likely in the field 30 years ago, this was unimaginable that we go from 3, 4, 5% of weight loss to beyond 20%. This was thought to be only possible with something like a gastric bypass. So it really is a breakthrough that we just, were unable to imagine just even a few years ago. There's superior impact here also on blood sugar that has partially to do with body weight, but not only. There is impact on insulin sensitivity that's independent of the body weight loss. There are, of course, some side effects in some parts of the treated populations, mostly early on, and predominantly some nausea, vomiting, and diarrhea that, however, subsides in many, if not most, of the patients after a while, and is not going beyond what you see with monoagonists that are on the market for GLP-1 receptor targeting. Now, this drug has really been approved last year in May for diabetes. It's expected to be approved soon for obesity by the FDA. It has been patented in 2015, and we had discovered it several years before that. Now, when you look at these biotech reports and industry reports, they're talking about the biggest drug ever. This is already outdated. I just read a report the other day of some science economists that are predicting $52 billion sales with just that drug alone worldwide. Now, it shouldn't be about the money, and hopefully these drugs won't be so expensive that they can't be used by all of those individuals that really would benefit greatly from them. But one more summary: dual agonism at the GIP receptor and the GLP-1 receptor significantly outperforms monoagonism. So 2 is more than 1, and that goes specifically for GIP, GLP. Well, if you can make 2 glucagon GLP, 2 GIP GLP, can you make 3? So we tried for a long time to come up with a triple agonist. GIP receptor, GLP receptor, glucagon receptor triple targeting was very difficult, took us many years back and forth. And here's the paper, finally published in 2015, that was achieved, was accomplished. And this is how any of these drugs basically look. It's a relatively short peptide. Every amino acid then is examined and exchanged or improved based on where you want it to be from, a GLP, a GIP, or glucagon, both or all 3. You need something like a fatty acid that helps you with albumin binding, that keeps it in circulation for a longer time. Talking about half-life, you want to keep it from being eaten up by protease cleavage immediately. And there is that CEX tail that was learned from the saliva of the lizard. Now, the data with the triagonist really blew us away. There's body weight loss that goes even way beyond of the dual agonist that you see here. That's a GIP, GLP. The triagonist is even better, going 25-30% weight loss. Now, keep that in mind. Food intake decrease also being similar. Improvements in fat mass, improvements in cholesterol, blood glucose normalized, insulin normalized. And if you now look at the very first clinical data that again come out of Lilly, the triagonist has impact and works in humans. These are phase 1 clinical data, phase 1b. When you put that in perspective with existing dual agonist data in humans, this is a tricky calculation and projection. It seems that the triagonist will be significantly more powerful, but still safe as compared to the dual agonist. We're talking about 50 to 100% better. Now, as a summary here, triple agonism outperforms dual and monoagonism. I can tell you there's going to be likely exciting new data of the phase 2 clinical trials of this first triagonist that will be disclosed at the American Diabetes Association in June. And we'll trying to see if this is, again, the same as we saw in preclinical data. That would probably mean, if it can be translated again, that we're looking at 25 to 30% weight loss with these triagonists in a safe manner over the course of about a year. Now, this is how the clinical pipeline looks worldwide. Several versions of dual and triple agonists by several companies going to phase 1, 2, and 3. So exciting times ahead. It looks like obesity becoming a treatable chronic disease where body weight can be normalized, similar to what we can do with blood pressure in most cases. I'm ending here so that Timo can have the chance to tell you exciting things about mechanism and the future of personalized metabolic medicine. But I do want to give you a take-home message that summarizes the work of 30 years. One, agonism and not antagonism of the glucagon receptor and the GIP receptor is what offers unprecedented potential for the fight against obesity and diabetes. Two, the gut hormone co-agonists, these new drugs surpass all existing treatments of obesity and diabetes preclinically and clinically. So that really it transforms the way we're going to be doing therapeutic applications for obesity in the future. And finally, key for the maximized benefits is that simultaneous activation of more than one human gut hormone receptor in the CNS, as we had predicted decades ago. So with that, I'll just thank everybody involved in this. This is just a sort of smaller selection of individuals that have contributed over the time, led by Timo Müller, led by Richard DiMarchi, Brian Finnan, and others. Many cooperation partners like Randy Seeley and Davide Calibiero, Diego Perez-Tilvi, and many others. And when you try to do this for decades and have a team that at times has 30, 40, 50 individuals, you need a lot of money. So I'm very grateful for all the funding we received along the way from Helmholtz, from DFG, from many other places like the Humboldt Foundation. And I thank you for your attention, and I'm looking forward to answer questions. If I understand correctly, questions will be answered after the second half of the talk that Timo will give. Thank you.
Dear Professor Choip, thank you so much for this amazing and very comprehensive overview of GLP, GIP, and glucagon receptor agonists. We all understand that we are only at the start of a real revolution in the field of obesity and metabolism. Therefore, excellent field for young endocrinologists and scientists to start working on. Manuel, my co-chair, Manuel Goethe, will continue now with the next excellent speaker, and at the end of the second lecture, we will have the opportunity to discuss all together.
Thank you, Lina, and thank you, Professor Matthias Schaub, for the excellent talk. Absolutely amazing. And now we have the opportunity to have Dr. Timo Müller with us. He will continue with this history. Dr. Müller studied the animal physiology at the Philipp University in Marburg, Germany, and in 2009 he received his PhD at the Department of Child and Adolescent Psychiatry and Psychotherapy at the University of Duisburg-Essen, also in Germany. He then moved to US, as most of us, to the Department of Metabolic Diseases, or the Institute of Metabolic Diseases in Cincinnati, and he returned back to Germany, leading the Division of Molecular Pharmacology at the Institute of Diabetes and Obesity. Since 2018, he is the acting director of this institute. As you probably know, Dr. Müller research is focused on the development and evaluation of novel unimolecular pharmacotherapies therapies to treat the metabolic syndrome, and in particular obesity and diabetes. He has published more than 150 manuscripts in top-tier journals that you all know: Cell Metabolism, Nature Medicine, Nature Metabolism, or Nature Communication. So we are sure he will provide us an excellent overview of these novel unimolecular pharmacotherapies. If you are ready, Dr. Müller, the floor is yours.
Yeah. Thank you very much, Manuel, for the nice introduction, and thanks for having me here. I hope you can see my screen. Yeah, so
yeah.
I could not have asked for a better introduction into the topic from Matthias, because this gives me a nice platform now to dive really into the mechanism of how these beautiful molecules that have been developed by Matthias Choip and Richard DiMarchi work in our hands. So quickly, here's my conflict of interest. I received from these companies support or a honorarium within the last 3 years. I also hold stocks at some of these companies, I have to say. And as Matthias mentioned in his presentation, the idea of pharmacologically decreasing body weight is not new. We see since the beginning of the 20th century a lot of drugs that have been used for the treatment of diabetes and for obesity. And what all of these drugs have in common is that they all decrease body weight in the single-digit range. Very rarely a molecule was able to cross the 10% threshold. And the first molecule that was actually able to quite safely cross the 10% weight loss threshold in humans was semaglutide very recently. And as Matthias alluded, we have a new kid on the block here, which is tirzepatide. And as Matthias mentioned, tirzepatide is an unbalanced GLP-1 receptor co-agonist with a 5-to-1 favor towards the GIP receptor. And what is known about GLP-1 and GIP is that GLP-1, next to the stimulation of insulin secretion, is that GLP-1 drives body weight loss by inhibiting food intake. But until recently, very little was known about GIP. And in the next couple of minutes, I will try to convince you that GIP is way more than a gastrointestinal hormone that is able to drive our insulin secretion. And when we look here at the SURMOUNT-1 phase 3 trials in non-diabetic patients with obesity, we see here that tirzepatide is able to decrease body weight by an impressive amount. Matthias has shown these data, 20.1% weight loss. This is far more than the single-digit range of weight loss that we have seen with previous drugs. And what is even more impressive is that we see actually in a substantial portion of these patients, up to 40% of patients, that we see a weight loss, a safe weight loss in the magnitude of more than 30%, right? Many of them actually even reach a weight loss of up to 40%, right? Which is very close to the efficacy of bariatric surgery. So for some reasons that we do or do not know, GIP is doing some magic to the addition of GLP-1 receptor agonism. And as Matthias nicely alluded, this is not intuitive because mice that lack the GIP receptor globally, they are resistant to diet-induced obesity. So what we see here on the left-hand side is what happens in wild-type animals that we feed with a high-fat diet. Expectedly, these animals gain body weight and get obese. And when we do the same with the GIP receptor knockout animals, we see that these animals, even so they get a high-fat diet, they have the same body weight as those mice that have been fed with a chow diet, right? With a carbohydrate diet. And this also translates to a significant improvement in glucose metabolism, right? In the wild-type animals that are fed with a high-fat diet, we see that as expectedly, these animals get insulin resistant, whereas the GIP receptor knockout animals are resistant to high-fat diet-induced insulin resistance. So there's some magic in also the antagonism of GIP, and this can also be used pharmacologically. Here we see actually a study from Novo, where they showed that when you combine a GLP-1 receptor agonist with a GIP receptor antagonist, you can get some degree of weight loss. But you note here that the amount of drug, the dose of the antagonist that you have to give is in the magnitude of 500 to 1,500 nanomole per kilogram. So this is a very, very high dose of an antagonist that you need to give in order to see beneficial effects of a GIP receptor antagonism. However, when you do exactly the same study with a GIP receptor agonist, you see here that a dose as little as 3 nanomole per kilogram is sufficient in combination with GLP-1 receptor agonism to drive synergistic weight loss. So while both the agonism and the antagonism of the GIP receptor, in addition to GLP-1 receptor agonism, drives greater weight loss relative to GLP-1 alone, we have to note that we need a substantially higher dose of the antagonist relative to the agonist in order to get beneficial weight loss greater than what we get with GLP-1 alone. So the first take-home message that I would like to drive home here is that GIP receptor agonism and antagonism can both decrease body weight when given together with GLP-1, but we need a substantially higher dose of the antagonism relative to the agonism, right? The second is that the mechanisms of how GIP receptor agonism or antagonism affect metabolism are largely unknown, right? And there are now several hypotheses that we want to talk now about. And the first is that GIP, for reasons that we don't know, affect metabolism. The second is that the coagonists might be just super potent GLP-1 receptor agonists, right? So maybe the GIP receptor is not relevant and the dual agonists are just better GLP-1 receptor agonists. The third hypothesis is that the coagonists are biased agonists for the GLP-1 receptor, right? So a biased agonist is a molecule that activates the GLP-1 receptor, but that causes different signaling relative to the endogenous ligands. Also, this is possible. So let's take this step by step. The first question, does GIP affect metabolism over unknown mechanisms? And in order to answer that question, we took diet-induced obese wild-type animals, and we directly injected an acylated GIP, a long-acting GIP receptor agonist, directly into the third ventricle of the hypothalamus. And what we see here is that a single injection of GIP into the hypothalamus increases CFOS neuronal activation, right? So CFOS is a neuronal marker that gets produced by the neurons when they get activated. And by showing here this increased CFOS, we know that GIP activates some neurons in the hypothalamus. We did not only see that GIP increased neuronal activation in the hypothalamus, we also see that when we give animals a single injection of GIP into the brain, that this single injection, central injection, decreases body weight, a single injection over almost 3 days. Right? And this is paralleled by a substantial inhibition of food intake. For the first 24 hours, the animals almost completely stop eating before the food intake then reverses to normal levels. So GIP somehow acts in the brain in order to decrease body weight and food intake. And here's some new data that are still unpublished, is that we show here that GIP given peripherally not only also increases CFOS neuronal activation in the hypothalamus, we see that a certain neuronal population of the CFOS activated neurons correspond to the POMC neurons. And we know that the POMC neurons are those neurons who get, for example, targeted by leptin to then inhibit food intake, right? So part of the neuronal population that gets activated by GIP are the POMC neurons, right? And when we chronically infuse GIP directly into the brain over a time of 14 days, we see that the GIP leads to greater weight loss relative to infusion of a saline control, right? And this is not seen in those animals that lack the GIP receptor in the neurons, in the brain, right? So apparently GIP needs the brain GIP receptor in order to decrease body weight. And the same is observed with the food intake. Also here we see that chronic infusion of GIP into the brain leads to a decrease in food intake. And this decrease in food intake is gone when you infuse GIP in animals that have no GIP receptor in the brain, right? So apparently GIP acts via the brain GIP receptor to decrease body weight and food intake. So we can go back to our hypothesis and we can answer the first question, is GIP affecting metabolism through unknown mechanism? And this is a clear yes. GIP decreases body weight and food intake via the CNS GIP receptor. Next question, is GIP or are the dual agonists just a super potent GLP-1 receptor agonists? And in order to answer this question, we worked here with a molecule that was also made by Richard DeMarchi. And here we have an acylated GIP. So a molecule that resembles pretty much the natural sequence of GIP. It is protected from the cleavage of the dipeptidyl peptidase 4 by an aminoisobutyric acid at position 2, the X here, and it carries a C16 fatty acid at the last lysine at position 42. It's pretty much the natural sequence of GIP protected by DPP4 and extended and acylated with a C16 fatty acid. And when we give this molecule into a wild-type animal, into a diet-induced obese wild-type animal, we see that this molecule nicely decreases body weight in these animals. And this is almost completely preserved in those animals that lack the GLP-1 receptor. So apparently GIP, in order to decrease body weight, does not depend on the GLP-1 receptor. But if the molecule depends on the GIP receptor, then we would expect that there is no difference in body weight. And this is exactly what we see. The molecule, the acylated GIP, decreases body weight in wild-type animals, but it completely fails to decrease body weight in the GIP receptor knockouts. So now you could argue, well, maybe this is a GIP-specific effect that is different for the dual agonists. And in order to see whether the dual agonists also depend on the GIP receptor, we did here a study where we compared a GLP-1, a balanced GLP-1 GIP receptor coagonist. So balanced means it equally activates both receptors at the same time with equal potency. And we compared this to a pharmacokinetically matched GLP-1 control, right? So it's the same backbone as the dual agonist. It just activates only the GLP-1 receptor. The half-life biodistribution is all the same. And what we see here in wild-type animals is that the dual agonist leads to a greater weight loss relative to the pharmacokinetically matched GLP-1 control. And when we do the same experiment in these animals that lack the GIP receptor in the brain, we see that the dual agonist now only works as good as the GLP-1. So the superior metabolic effect of the dual agonist to decrease body weight disappears when we eliminate the GIP receptor in the brain. So the dual agonist needs the brain GIP receptor in order to drive greater weight loss relative to a pharmacokinetically matched GLP-1. So at this point, I think everybody would agree that GIP matters for the metabolic effects of the dual agonist. I think nobody would disagree here. But now we have some interesting data from Lilly, and they used their dual agonist, which is even an unbalanced dual agonist, as Matthias mentioned. It's a 5-to-1 dual agonist that favors the GIP receptor over the GLP-1 receptor. But when we give GLP-1, or when Lilly gives tirzepatide into wild-type animals, they see a substantial decrease in body weight, which is gone in the GLP-1 receptor knockout animals. So in contrast to Marcadia 709, tirzepatide apparently drives weight loss only over the GLP-1 receptor. So how do we explain this? And this is actually easy to understand if we look at the native peptide. The native human GIP, and this here is data from Jens Holt from Copenhagen. And what he shows here is that the native human GIP is a very weak and only partial agonist at the mouse GIP receptor. So human GIP is not able, irrespective of the dose, to fully activate the mouse GIP receptor. And tirzepatide is based on the human GIP sequence. So we would expect that tirzepatide, despite being a dual agonist in humans, works only very poor at the mouse GIP receptor. And this is actually data that is in press. It's a collaboration between Novo Nordisk, Lilly, us in Helmholtz Munich, and John Campbell in Duke. And what we show here is that when we take tirzepatide and we look at the GLP-1 receptor signaling, G-alpha S recruitment, in cells that express the human GIP receptor, we see that tirzepatide is equally potent as human native GIP to induce GIP receptor signaling. But when we do the same experiment in a cell that expresses the mouse GIP receptor, we see exactly the same. We see that tirzepatide is only a very weak and partial agonist at the mouse GIP receptor. And this has, of course, fundamental consequences when we look at the mode of action of tirzepatide in mice. And here are also data from this publication that is currently in press. Here we see that human GIP is 10-fold less potent at the mouse relative to the human GIP receptor, that tirzepatide is 75-fold less potent at the mouse relative to the human GIP receptor, and that Marcadia 709, the dual agonist developed by Richard, is only 2.6-fold less potent at the mouse relative to the human GIP receptor. The consequence of this is that tirzepatide is not the ideal dual agonist to assess the mode of action of GLP-GIP coagonism in a mouse. And this message is so important that I also want to show you some data that is currently in press. And here actually we are looking at isolated mouse islets that are treated with either a GIP receptor antagonist or a GLP-1 receptor antagonist. And then we give, after pre-treatment with the antagonists, we give tirzepatide. And what we see here is that tirzepatide-induced insulin secretion is fully preserved in these islets after we antagonize the GIP receptor. But when we antagonize the GLP-1 receptor, tirzepatide fails to stimulate insulin secretion. So in the mouse islets, tirzepatide stimulates insulin secretion almost exclusively via the GLP-1 receptor, right? But when we do exactly the same study in human islets, the situation is completely different. Here, tirzepatide-induced insulin secretion is largely decreased when we antagonize the GIP receptor, but is almost completely preserved when we antagonize the GLP-1 receptor. So the conclusion is that tirzepatide is primarily a GLP-1 receptor agonist in mice, but a very potent dual agonist in humans. And this has, of course, fundamental consequences when we look at manuscripts that use tirzepatide to assess the mode of action of dual agonism, because we don't sufficiently activate the GIP receptor in order to find meaningful effects of how the molecule works. Okay, so to answer the next question, are coagonists just super potent GLP-1 receptor agonists? The answer is a clear no. GIP shows preserved weight loss in GLP-1 receptor knockout mice. Marcadia 709 loses superior potency over GLP-1 in the CNS GIP receptor knockout mice. And tirzepatide needs the GIP receptor in humans to affect insulin secretion. But we also have to note that tirzepatide is primarily a GLP-1 receptor agonist in mice, but a full dual agonist in humans. Okay, next question. Are coagonists biased agonists at the GLP-1 receptor? And in order to answer this question, we see here a live cell imaging of a GLP-1 receptor positive cell that expresses the GLP-1 receptor and is treated with native GLP-1. And what we see here is the movement of the ligand receptor complex inside of the cell, right? So the internalization process. And as you appreciate here, there is a lot of dancing going on the cell, indicating that there is a high degree of ligand-induced receptor internalization. The same is observed with semaglutide. It's almost indistinguishable from the native GLP-1, but both dual agonists show a significantly delayed rate of receptor internalization, indicating that the ligand stays longer on the receptor on the cell surface to prolong receptor signaling. So the key question is, is this important for the metabolic effects of the drug, right? And in order to answer this question, and this here is actually the data that are picked from the movie, we see here that tirzepatide shows a significantly delayed rate of ligand-induced receptor internalization via the GLP-1 receptor. And now we made, together with Novo Nordisk, a molecule that is based on the sequence of semaglutide, of the blue molecule, and we modified the semaglutide, the blue molecule, in a way that it mimics the internalization rate of tirzepatide, right? So we call it a beta-arrestin biased semaglutide, right? And what you see here is that when you compare now both molecules, tirzepatide on the left-hand side and the beta-arrestin modified molecule on the right-hand side, that the rate of receptor internalization is now pretty comparable between tirzepatide and this beta-arrestin modified semaglutide, right? And we also looked at beta-arrestin 2 signaling, and we see here on the left-hand side that tirzepatide shows a significantly decreased rate of beta-arrestin recruitment, and this is now also seen with the tirzepatide-like semaglutide, right? And also G-alpha-S recruitment is substantially decreased by tirzepatide and comparably decreased also now by our beta-arrestin modified semaglutide. And we also looked, of course, for cAMP. We see here that tirzepatide is a little bit less potent as semaglutide to induce cAMP production. And the same decreased potency to induce cAMP production is now also seen with our semaglutide beta-arrestin-like molecule. Now, and the key question is now, how does this transfer to the metabolic effects of this drug? And we compared now semaglutide against the new semaglutide, the beta-arrestin modified semaglutide. And we appreciate here that the modification of semaglutide towards a beta-arrestin biased signaling further decreases body weight compared to semaglutide. And this is not only seen for the body weight, this is also observed for the food intake, right? So by modifying the sequence of a pure GLP-1 receptor agonists, we can further drive weight loss. And this is probably contributing to the metabolic efficacy of tirzepatide. So tirzepatide is a better GLP-1 receptor agonist, but on top of this, we have the beneficial effects of the GIP receptor agonism, right? So we can now also answer the third question, which is that are dual agonists a biased agonist at the GLP-1 receptor? Yes, that's the case. Both dual agonists are biased towards GLP-1 receptor signaling, which probably further enhances weight loss and further inhibition of food intake. And what we are now currently doing in our lab with support of the European Research Council is that we are mapping the GIP receptor network. We are identifying the brain regions where the drugs end up and mediate the effects of GIP receptor agonism and antagonism. We are delineating the mechanisms that underlie the regulation of metabolism by GIP receptor agonism and antagonism. And we are validating the function of targeted loss of GIP receptor signaling in certain brain areas. And I want to give you as a last slide a little teaser of our recent work. And here actually we eliminated the GIP receptor from a very small population of neurons in the brain. And what you see here is that we injected these animals with a cyanine 5 labeled GIP, so a fluorescently labeled GIP. And this GIP molecule heavily reaches the area postrema in the hindbrain and causes C-force neuronal activation in the hindbrain. And this is eliminated, completely eliminated, or almost completely eliminated in those animals that lack the GIP receptor in this specific set of neurons. And what is really interesting is that when we give GIP, this acylated long-acting GIP into wild-type animals, we see that the GIP decreases food intake in these wild-type animals as expected, but this is no longer observed in those animals that lack the GIP receptor in these certain neuronal populations. So to make a long story short, I think we have identified the neuronal population that drives weight loss and inhibition of food intake by GIP receptor agonism in the brain. And with this, I would like to close. The people that I have to thank are, actually, there are so many people I have to thank, they don't fit actually on the screen. But most importantly, here are Matthias Chöp, Richard DiMarchi, and Brian Finn. And last but not least, also the companies who didn't give up on us over the last 15 years and supported us here financially. And with this, I would like to thank you all for your attention, and we are happy to take your questions.
Thank you, Professor Miller, for the amazing lecture and for your contribution in the field. Your work is really, interesting and amazing, really. The more interesting the lectures, the more the questions, and we have a lot of questions from the audience. First one, are you afraid that these drugs lose efficacy over time, or you think that this is not a threat?
Matthias, do you want?
Yeah, should I get started? We have no data, and there are no clinical data indicating that they would lose any of their efficacy. That said, current thinking would say that these are drugs that have to be taken lifelong. The more experience we will have with them clinically, the more different of these dual and triple agonists will be approved, the more we will learn. I'm assuming at some point there will be a version of a maintenance therapy that may involve other drugs, may involve leptin, and just the initial weight loss therapy will be driven by these dual and triple agonists. That would be my prediction, but the answer is no. There's no indication that they lose efficacy over time.
Thank you. Another question that follows the first one, how long we should continue these medications if we reach the target of losing of our body weight? Should we continue? Are they drugs that should be taken for the rest of our lives? Is like antihypertensives. If I take the antihypertensive, I don't have hypertension. This is the same with obesity and anti-obesity drugs?
Timo, why don't you take this one?
Yeah, so very happy. So actually the beauty of the GLP-1 receptor agonists is that the weight loss is even greater the more fat mass you have to lose, right? So after you have lost a significant amount of body weight, right? Let's say 30% or something, right? Then the efficacy of these drugs is very limited to drive further weight loss, right? So at some point, when you have no more fat mass to lose, the drugs will not drive cachexia. We have not seen in our studies, nor is this published in the literature, that you can dose GLP-1 receptor agonism so high that you will drive cachexia. There are studies actually in rats and even in humans saying that the weight loss efficacy is lower the fat mass of the patient or of the animal model is, right? So it's very unlikely that we will drive cachexia.
So the answer is that we should probably continue as a maintenance therapy, or we should stop the medications at some time point.
I mean, if you stop the medication, the patient will regain body weight, right? So as Matthias said, it is likely a lifelong treatment, right? I'm just saying that the degree of weight loss will at some point stop, but you still need to take the medication to prevent the weight gain, right?
Okay, thank you. There is another question about cardiovascular safety. Of course, we know that we have some cardiovascular benefits, but would you like you both to comment on that?
Matthias?
Sorry, can you repeat the question?
Someone asks about cardiovascular safety. We already know that we have benefits, not only safety, but we would like to hear your opinion.
Well, the indications are currently that there's absolutely safety. Second, there may be benefits. Now, cardiovascular endpoints to be studied, that's a long game, and there will be additional studies that need to be done. But indications now are that there is benefits that go beyond what we have seen with monoagonism, and that probably has to do with three things. One, the weight loss, so there's healthier metabolism, less fat mass, and with all the problems that come with fat mass that now aren't there anymore. Second, GLP-1 receptor benefits that are known and described that happen through direct presence of GLP-1 receptors in the cardiovascular system. And third, GIP and glucagon receptor, or in the case of tirzepatide, GIP receptor mediated benefits that may, for example, directly go through liver lipid changes or changes in circulating lipids, cholesterol, etc. And that will potentially be yet another layer of benefits that protects from atherosclerosis, etc. Timo, did I forget something?
No, I totally agree, right? I mean, there are 5 very big cardiovascular outcome trials of GLP-1 receptor agonism in high-risk patients, right? So patients that are at risk for a cardiovascular event. And all of these 5 cardiovascular outcome studies show that GLP-1 receptor agonism is safe even in high-risk patients. The greatest side effect cardiovascular-wise is a very small increase in heart rate of 2 or 3 beats per minute. But this is not really concerning, given that these drugs have a significant improvement in life expectancy, the risk to develop a heart attack, a heart disease. And similar things are also meanwhile reported for tirzepatide, right? So there is one study in Nature Medicine, I think, that looked at the cardiovascular benefits across all the available phase 3 trials. And they see exactly the same as what has been reported for the cardiovascular outcome trials of GLP-1 receptor agonism. And also in the literature, there's no really good argument that would argue that the GIP receptor agonism would have detrimental effects on the heart.
Thank you so much.
Thank you. Mm-hmm. There are a couple of questions regarding the long-term effects of this agonist in several different endpoints. One of the question is asking about the continuous activation of those receptors. Is this could be detrimental in some way, or the putative effect on insulin secretion? If long-term treatment with this agonist could have some effect on insulin or in another parameters that we didn't test or we don't know. What do you envision about that?
Matthias?
Yeah, I can maybe start. I mean, first of all, any drug is extensively studied before it gets FDA approved. And of course, things like insulin secretion, etc., has been very much in the focus of what had been examined by the FDA and in clinical trials, up to the point where tirzepatide Mounjaro has been approved by the FDA last year. And now there's additional data. So it appears to be absolutely safe. Now, of course, these drugs have not been tested for 10 years or 15 years yet, but there's extensive experience in millions of patients with monoagonism. And that is very comparable in many ways, stimulating insulin secretion. The pure sequence of these hormones is very similar, and there's no indication that there is any unexpected long-term issues. But so very, very, very unlikely, and it seems to be very safe. As a matter of fact, dual agonists or triple agonists potentially might have even less side effects than monoagonism, but no data that indicate any trouble here.
If you allow me, the question comes from a very experienced professor, from Professor Kalliopi Kotsa from Aristotle University of Thessaloniki, and I'm glad to see her with us. And I'm thinking that she asks about the stimulation of the cells, the continuous and chronic stimulation of the pancreatic cells. Are we afraid of this stimulation of, in terms also of proliferation of the cells, cancer or things like that?
I can start, Timo, but you should chime in. Now, if this would be a cell that would not naturally be stimulated via that pathway, we could be philosophically at least more concerned. But now these cells are being made to constantly being stimulated by these pathways. Every time there are changes in nutrient intake or lack of thereof in the gut, there are changes in these gastrointestinal hormones that then go and stimulate these cells every day, every hour of our lives. Now, there is an adjustment, and it's a philosophical perspective. If you want to say we, with these drugs, are normalizing what should really be higher, or we are going slightly supraphysiological because the system became a little bit resistant. Think about insulin that's given lifelong and to overcome insulin resistance. Now, but not going into these details, that stimulation happens anyway throughout lifetime. That's not an artificial thing per se. The question is how much of that in addition and what it does. And there seems to be no indication that that's a problem. Timo, anything to add?
No, it's absolutely right. I would actually even argue in the opposite way. I would say actually that these medications help the beta cell, right? Because in an untreated diabetes, right, the beta cell is suffering what we call glucotoxicity, right, which really harms the beta cell, right? The beta cell gets inflamed, and they hypersecrete insulin, and then at some point the beta cells just give up and die, right? And GLP-1 not only basically improves insulin sensitivity in the beta cells, it also is anti-inflammatory and stimulating proliferation, at least in rodents, right? And this actually helps the beta cell to survive. Actually, even under conditions where the beta cells die, usually the molecule that makes the GLP-1 is the pro-glucagon, right? And you need the pro-hormone convertase 2 in the alpha cell to make glucagon and the pro-hormone convertase 1 to make GLP-1. But in the beta cell, right, when the beta cell is dying, the alpha cell switches from pro-hormone convertase 2 expression, which makes glucagon, to produce GLP-1 by switching to production of pro-hormone convertase 1. And the reason why the alpha cell is doing this is because the alpha cell then produces GLP-1 to help the dying beta cell to proliferate and to make more insulin, right? So the GLP-1 system is a very beneficial, positive system. And we know from a lot of clinical data, Matthias just mentioned this, the first GLP-1 receptor agonist was approved by the FDA in 2014. And we have patients on this medication since now almost 10 years, right? So we have a lot of clinical data that all argue that the constant stimulation of the GLP-1 receptor system is not detrimental for glucometabolic health.
Mm-hmm. Do we have some inputs from the preclinical models? Because with mouse, we don't need to wait for 20 years or 15 years, so we can see it in a short period of time. Do you have those data on animals?
Oh yeah, of course. I mean, the problem with the mouse models is a little bit different because the mouse islets, they proliferate pretty friendly, right? And which is way different to the human islets, right? Mm-hmm. So the human islets, if they double within 1 week, you have a good beta cell, and the mouse islets proliferate in 24 hours, right? But we know from a lot of rodent data, and even in human data, we have human data that shows this too, that if you have a good system and a good donor of the human islets, then you see that GLP-1, constant GLP-1 receptor agonism stimulates proliferating of the beta cells, right? And we have even drugs that are made to target beta cells under conditions of type 1 diabetes, because even in type 1 diabetes, you still have a couple of beta cells left, right? To help the beta cell to proliferate and to decrease glucotoxicity and inflammation.
I have to leave a little early, but Timo can answer all the other questions, I have no doubt. It was great contributing here.
Thank you, Professor. It was great to have you with us. Thank you for everything.
Thank you so much for being here. So there is another question that I think is perfect for Timo, that is regarding the putative effect of these compounds on autophagy, and if this can be some way affecting cell homeostasis.
Yeah, so I mean, GLP-1 works anti-apoptotic and anti-inflammatory, and it inhibits actually autophagy, right? So at least in the beta cell. So it helps the beta cells to stay happy and to survive longer, right? So we have no data that currently indicate that the GLP-1 mimetics have any detrimental effects. The greatest side effects, other than the gastrointestinal side effects, is a small risk of gallstones, right? But this is a very, very, very tiny percentage of patients that have this. And even the gastrointestinal side effects are very transient, and they typically disappear in the majority of patients within the first 4 to 6 weeks. And this is also the reason why you stepwise increase the dose of these medications, right? So tirzepatide actually had a 4 to 8 week stepwise increase in the dose so that the patients don't have too many gastrointestinal side effects.
Okay.
Thank you. I think that there are no more questions. Manuel?
No, I don't think so. I think we asked all the questions that we received from the chat, and there is no live questions right now. No one waiting for the connection. So,
and People thank you for the presentations and, of course, for your original research and your contribution in the field. We would also like to thank you all, to thank the two speakers for the excellent lectures. All of you for the active presentation. I think that this was a very live, webinar, and this is what we aim for. The topic is hot. The field of metabolism and obesity is amazing and very promising, and a good one to go for young endocrinologists and scientists. We look forward to welcoming you in the next Spotlight on Science. It will take place in 1st of June. So mark your calendars and look forward to meeting you all again. Professor Miller, thank you again for your presence.
Thank you
so much.
me. Thanks for having me.
Bye-bye.
Bye-bye. Thank you so
Have a
much.
nice evening.